Okay. And just as a reminder, if you go to the due dates, you will notice that some of the urinary stuff is due next week, and some of it's due not until November 1st. So let's see, let's pull that up real quick. Okay, so we're on week 9. All right, on week 9, what we're going to focus on is just the anatomy portion of the urinary system. So with that said, you've got a kidney dissection that you're going to put in the drop box on October 25th. So we're going to go over that, okay? And then the following week, we're going to finish up with urinary case studies. We won't get to the case studies tonight, but what we'll do is we'll go over them next week, and then they'll be due November 1st. So we'll talk about acid and base balance, more of the physiology of the urinary system. We'll mention it a little bit tonight, but we're not going to go over that yet. We're just going to hit the anatomy tonight. So we'll just talk about some very basic things related to the physiology, and then we'll focus into that next week. So for tonight, we're on week 9. You guys are going to have a post lab that's going to be due. Of course, you're going to get on, look at the images through the dissection tool, through Connect. And then you guys will have post lab dissection to do. And then don't, like I said, don't do the case studies. We'll do that. We'll get into that next week. Okay. All right. So that's where we are. Now, let's see. We are on chapter, come down, what is it? 23, 24, 23. Okay. So we'll come down. All right, so here we are, Lab 7 urinary system. Here we are. We're going to go over the objectives. So the objectives, some of them, like I said, we're going to go over some of it tonight, some of it next week. So tonight we're going to describe the function of the urinary system. We're going to talk about it very basically. We're going to look at the structures. We're going to spend a lot of time on the structures because the urinary system is very detailed in how it works. We'll talk about some basic physiology, but we're not going to talk about what happens when this physiology or the pathophysiology, these abnormalities happen. So we're going to focus on that next week. Then, of course, structures, regions of the dissected kidney. When you do the dissection, we have specific instructions on that. So that's, like I said, it's due October 25th. However, I'm going to spend time going over that once we get over everything, because there are things that you need to pay attention to, okay, that I'm going to talk about because it counts for points, okay? So we'll come back to this. Locate, identify, selected structures, urinary system on a dissected fetal pig. Well, we aren't there yet. So we'll come back to that. All right, trace blood flow, filtrate through the kidneys and the nephrons. So the nephrons are the little basic units that make up the kidneys. We're going to talk about those. We're going to look at a nephron. You guys are learning the structure of the nephrons and how the blood flows through to create the urine, okay, and how specifically that happens. And then the next one is list physical characteristics of urine, including the pH and specific gravity. That's something we're going to focus on next week. List normal and abnormal urinary constituents. That's going to be next week also, okay? We're going to mention it a little bit. So we're just going to talk about what's considered waste, if it builds up a little bit. Next week, we'll talk about that in detail. And then the next one says name conditions associated with abnormalities. That's next week. Conduct selected urinary tests. That's next week also. So keep in mind, we're getting through most of the objectives. And then by next week, we're focusing on the pathophysiology of basically what if we're interpreting lab results, we're looking at things that are, abnormal, what's considered normal. That's all next week, okay? All right, so tonight we're going to go over the handout like I normally do. You guys, of course, have the dissection tool that you've got a lot of things to identify. However, like I always do, I try to go over everything with you. But if you scroll down here, here's under, it's telling you, here's that, dissection tool you've got to go to, and then here's all these things you've got to be able to label. If I scroll down, you're going to see that there's going to be a lot, okay? But everything's very differentiated from one specific thing to another, meaning that everything's going to look different. So it's going to be easy to identify all of these different structures. Okay, I don't think that it's hard. In my opinion, I don't think it's hard. I don't think students have had trouble with it in the past. Let's put it that way. Come down here, there are some specific things you need to look at, like overview portions of the urinary tract. Make sure that you spend time looking at all of these things. If you pull it up in the dissection tool, there is also where we've posted or placed this nephron drawing practice sheet where you guys can go in, you can color it, you can label it. Let me just pull it up real quick so you can see it. This is something that's from the lab manual. I'm going to be honest about it. I think it's great for studying and labeling and getting things down. I think it's for your benefit. That is nothing that you're going to actually turn into me, okay? So the little practice sheet is completely for you. If you want to color it, you want to label it. Once you kind of understand the whole concept of what this is actually, then you guys can have that for your own use, okay? All right, backwards. Of course it was going to throw me out of that. Okay, hold on. Let me get back. I still struggle sometimes with that. All right, so below that is going to be the dissection. I'm getting there just to show you. There's very specific things about the dissection. So I'm going to mention it. We're going to go back to it when we're completely done, so you guys don't have any confusion about it. But I do want to mention it just in case if somebody leaves class or whatnot. Okay. So here's all the identification off the dissection, cadaver dissection tools. Go down here, this is where we were. That was the drawing that you can click on and you can use it for your own use. It's for practice, and it says here even, not required to submit this, okay? Now, next is dissection instructions, okay? You actually have a video that goes along with this, meaning it shows you specifically what you need to be labeling when you do the dissection. However, that is a guide to help you. That is not you doing the activity. So, and that is not to take literal, let's put it that way. What I want you to do when you get to this portion, because this is going to be due October 25th, when you get to this point and you have the free time to actually spend time on this, you need to read through every bit of these instructions. Now, we've already done a dissection with the heart. You guys probably noticed you may have gotten points off of things that were like, you know, I didn't make it in a PowerPoint, the pictures, I didn't make the images real clear, you know, I didn't get the labels just right, you know, with the pens, things like that, or I didn't spell the actual structure correctly. All of that counts for points, things that you may not think about being online and things that we need you to do. Specifically to make sure that you're actually following through with the complete activity. We need you to do all of it, because if you don't do all of it, you don't get all credit, okay? But with that said, follow this specifically. And what I mean by that, it tells you what to do just to get ready. I don't need to read through that. But when you start, it tells you, okay, go ahead and label your pens, small pieces of paper. Obviously, you're going to take images. They need to be clear. Label the following structures. Take the photo with them, okay? Each photo should be close up. Clearly see it, okay? Put one pen in a photo. So you guys, last time, you would do like several pens, and it would be a general big, you know, back up to get the whole thing, right? And then it was hard for us to be able to see specifically. Maybe we were able to tell bigger structures than the smaller things, like the valves sometimes were hard to see. Even like when you guys labeled chordae tendineae or something like that with the heart, it was hard to kind of tell, like, is that where that pen's going? I can't really tell from that image, you know. Keep that in mind. Try to put yourself in our perspective, looking at those structures, and you don't even know what the structures are. Does that look like that makes sense? Okay, think about who's viewing that, okay? Then here, each photo needs to have a card next to your kidney with the name and date. See, those are things that you're going to get counted off points if you don't do it correctly. Kidney structures to pin, you don't have a lot, thank goodness. You've got 7, okay? It shouldn't be a big deal. You need to number them. Make sure that they correlate to what it's supposed to be. We've had some people mess that up in the heart dissection. Trust me, we have. And then here's one thing I want you guys to focus on, because you're going to get points counted off if you don't do this specifically the way we ask. All photos need to be in a, look, JPEG, look at the JPEG here. These are the different formats. All of the photos need to be in one of those formats, okay? Upload the photos, put it in Dropbox, okay? This tells you exactly what to do, all right? If you mess up, you skip a step, you don't do a step, something doesn't look right, you should literally, what I would do personally is print this off and go check mark, I've done this, check mark, I've done this, check mark, until you complete the whole activity, okay? Obviously, we're not gonna know where you threw your gloves and things like that you cleaned up. We don't care about that. I mean, we care about it, but we're not grading that, right? We're just giving you some extra tips, mindful things that, you know, please wipe up good, please we're giving you little instructions here just to help you. But the things that the actual thing you were turning in, the pictures with the labels, with the pins, with the right photos, all of this specifically needs to be on target. So you need to go through each thing, check mark, just like I told you guys, the name and the date being on there is something that counts for points, okay? So please keep in mind when you go through this activity, follow the instructions to detail, like just every single instruction, run through it. Don't skip it. Don't come down here and say kidney structures depend, here's the photograph, here you go, okay? That's not what we asked for. And make sure that it goes in the Dropbox, okay? I'll come back to that if you guys have any questions. We are not getting tonight, like I said, here's the physiology portion with the urinalysis. We are not getting to that tonight. We'll get into that next week. Don't jump into this. Please don't jump into this yet until next week. You guys do have a post-lab urinary quiz. It's not due for some time. Like I said, all of this is going to be completed, but from tonight, next week will be the full completion of the whole urinary system. So just give us some time to complete everything. I don't want you guys to be overwhelmed to look at this and think you have to do all of this. All right. That's why you always go back to the due dates. All right. With that said, now that we've looked at everything there, and we'll come back to the dissection if anybody needs me to, let's skip over. We're going to go to the handout. And let me make this, hold on. I'm trying to make this a little bit bigger because one of the images doesn't, well, it's probably big enough. One of the images is not awesome. Let's put it that way. It's not awesome when I scroll down and it's just not big enough, but we'll get there. Okay. So urinary system functions. What is its overall function? We talk about the urinary system. Major function is we want to get rid of nitrogenous waste. Okay. Of course, what does that mean? We're going to talk about what do we consider nitrogenous waste. You guys have probably heard of things like uric acid, urea, your creatinine levels. You probably you've had them tested, I'm sure at some point in time, if you've hit a certain age, somebody has probably tested your kidney function at some point in time. Or somebody that's had kidney issues, maybe kidney failure, maybe it's from being, the most common type is from diabetes, for example, okay? But when we look at the kidneys, we want to think about it is going to be a filtering system. That is the basis of it. But when we filter our blood and result to create the urine, we're maintaining electrolytes, acid-base balance, and fluid balance for the blood pressure, okay? So it is a major, and this is what's listed here, a major homeostatic organ, okay? And not to say all organs don't play a role in homeostasis, because all organs do. But this one's a major one, okay, because it's filtering the blood. We've got 2 kidneys, we can live with one, but clearly, we intended to have 2 evolutionary wise because it's smart. If we lose one, we can still, function with the other. All right, so whole function is to filter the blood, get rid of the waste, okay? Toxins, metabolic waste. So metabolic waste is going to be the byproducts of metabolism. Excess ions, if we need to get those to get, you know, rid of them, because we can cause different problems in the electrical balance, right, of our blood. Allows retention of needed substances, and what needs to either be removed or returned to the blood. This is supposed to be blood, guys, if you want to make a note. Malfunctioning urinary system. What happens? So if your kidneys don't function properly, eventually it will lead to death, right? But kidney failure, what happens is we're literally being poisoned. So you're having your urea, uric acid, creatinine levels, all of these byproducts, okay, of metabolism, breaking down proteins, okay, all these different things that are happening that should be waste and that we need to get rid of, no longer can we get rid of if the kidneys don't function properly, right? So this will be fatal. So you're literally, it's poisoning yourself if we can't filter out the blood properly. So when we look at the kidneys, and I know you guys know where the kidneys are, but we have a pair of kidneys. We're all typically born with a pair of kidneys, right? And then you've got these little tubes that come from the kidneys called ureters. They transport the urine from the kidneys down to the bladder, okay? And the bladder is just going to be a storage reservoir, okay? And you guys remember we talked about epithelial tissue, and we talked about the bladder. We talked about transitional epithelium, and transitional epithelium allowed for the bladder and part of the ureters to expand, okay, and recoil back, right? And the whole basis of that was it's a storage tank. And if we get to certain levels in that storage tank, we can, so we don't urinate on ourselves, we can hold it till we're ready to go to the bathroom. That's the whole purpose of that transitional epithelium. And then when we look at the bladder, how does the urine get released? Well, it gets released via a tube called the urethra, okay? All right, so we'll come down here, and you guys are gonna label the way your handout's done. You can label all the different structures. So here's the kidneys, right? These are the adrenal glands. And then you can see the renal artery, the renal vein, okay? Here are the ureters that go down to the bladder. And then if you come down here, here's the urethra, okay? And all of these things on these images, by the way, as we go through them, they are actually labeled like up here. You just bring them down and put the labels with them. All right. Okay, now I'm going to skip over some stuff because it makes more sense to me as I go over it for you guys, all right? So since now we've looked at the kidneys, why don't we look at the general structure of the bladder, the urethra, and the general structures that differ ourselves, females from males or males from females. Let's start with the female. Okay, so here are the ureters. Here's the bladder. Okay, and this is no bigger than the males. It's just showing it in the bigger image here. So here's the ureters. Here's the bladder. You've got this good muscle tissue around the bladder. It's going to help contract and push out urine. You'll see the term up here, detrusor muscle, that's it. And then here's the urethra, okay? When you look here, you're gonna see these little openings. These are these little openings or orifice that's allowing for the ureters to open up into and bring the urine into the bladder. If you look in the back, and this is not something you have to label, but we call this like a rugae. So it's like an irregularly shaped. And that makes sense because it's transitional epithelium, right? We allow for it to plump up when it's empty and then expand when you fill it up with urine. Okay, and then right through here, and it's actually labeled here as a triangle if you look at it, but we call this the trigon of the bladder. So the trigon is where we have the three orifices. So we have the orifices where the ureters come in and they open up to bring urine. And then if you come down here, here's the third orifice that's leading into the urethra, okay? And it creates kind of like a triangle. So we call it the trigon, okay? And then if you look in the female, the urethra is very short. So it's just a little over an inch, for example, okay? And then we'll come over here and look at the urethra in the man in a second. And then this is the external urethral orifice in the female. So You know, I don't want to say there's not a lot going on, but for the urinary system, there's not as much going on over here as the male, right? So over here, here's the ureters, there's the detrusor muscle. Here we go, we've got that trigon structure, the rugae in the back. And then as the urethra goes down, it's inside of the prostate right now. This is the prostate, guys. So inside the prostate, we give it the name prostatic, or prostatic meaning the prostate, urethra, okay? And when it comes down and gets to the penis, we call it the penile or spongy urethra, okay? And then this, right, we don't have it labeled here, but this is the intermediate urethra, okay? Or sometimes we call it urogenital urethra, but just because where it meets. And then down here, we have the external urethral orifice, okay? So not a ton we're looking at from the cross section, but it's enough to give you an idea. I mean, of course, some of these images we'll get back into, especially with the male, that has to do with reproductive, like the glands here, the prostate, for example, and specific things about the penis, okay? So, all right, come down. We're gonna skip over this. And then we're gonna skip over this too. And we're gonna come down to the kidney. This is the image that it's hard for me to kind of make it big enough for you guys to see. This is the one I was kind of debating with how to get this bigger. Okay, so what I want you guys to first see, hold on, let me try to make it the best of my ability in the screen, okay. So what I want you guys to see with the kidney is we want to go over the structure. So here's the outer portion, the fibrous structure of the kidney. Notice it's a good red color. This portion right here is the cortex of the kidney. We're going to talk about what all these little things are, the arteries or arterioles and the venules and veins. We're going to specifically talk about those, but this is all the cortex. Okay, it looks like a little bit of, I don't know, really pretty fireworks out here. And then when we get inside, this is considered the division line from the cortex to what we call the medulla. Here it's even labeled there. This is the medulla. So we call these specifically, each one of these is called a renal pyramid. Okay, these are renal pyramids. Okay, and they're very striated, but we're going to get to that in a second, why they're striated. So these are the renal pyramids inside the medulla. These are the columns that divide up the renal pyramids. So here are the columns. And then when you get down here, because once you get to the apex or the papilla, sometimes we'll call it papilla or the apex of the pyramid, we call this a minor calyx. And you guys have all of this labeled, by the way, in your book. I'm mentioning it, even though it's not labeled here. This is the minor calyx. When it gets bigger, we call it the major calyx. So, for example, here's another minor calyx, major calyx, minor calyx, major calyx when they meet together, minor calyx, major calyx, minor calyx, major calyx, okay? I miss my pointer in class sometimes. Okay, so this is all considered the medulla, all the medulla. This is the minor calyx, this is the major calyx, and now we're going to have, all of this is going to be urine where it's going to drain in, but we're going to talk about that here in a second in more detail. Urine's going to come down here into what we call the renal pelvis here, and then it's going to drain out into the ureter down to the bladder. Okay, now how does this really work? So we just talked about the general structures of the kidney, but how does it actually work? So what happens, all right? Urine is created from your blood, okay? Now let's explain how this works. So you have your renal artery. Your renal artery is here, okay? And it comes in, so blood gets supplied into the kidney, and it goes into segmental arteries that get branched off, okay? You see it branching here. Then it goes into interlobular arteries. So here it's branching off interlobular arteries. One would go here, 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 and here, okay? Then once we keep continuing from the interlobular arteries, we create something called the arcuate artery, okay, creating an arch, an arcuate artery. Here we go. These are arcuate arteries. These are at the base of the pyramid, so arcuate arteries, okay. And then from the arcuate arteries, we have little branches. You guys see these little tiny arteries branching out into the cortex, a little red branching out, looks like little trees. Those are called the cortical radiate arteries. They radiate out. So they're of the cortex and they radiate, okay? These are the cortical radiate arteries. Now, what's coming next, you cannot see, but I'm gonna pull that up in an image in a second. So if you follow your diagram here, it says the cortical radiate arteries go into the afferent arteriole. Remember, afferent means going towards, okay? Afferent arteriole, you can't see this, but I'll show you in an image in a second, okay? The afferent arteriole is supplying and actually supplying what's considered the nephron, okay? This is all considered within the nephron. Okay, all of this is boxed off, for example. All of this is boxed off because it is considered inside or supplying the nephron, okay? The smallest portion or subunits of the kidney that allow for it to filter and create urine, all right? So we're gonna look at this in a second. It's the afferent arteriole to something called the glomerulus, okay? Glomerulus meaning one, glomeruli meaning more than one, plural, and it's a capillary bed. We're gonna look at that capillary bed. From the glomerulus, we have a efferent, meaning exiting, right? Exiting, so efferent arteriole, and it creates something called peritubular capillaries or vasa recta capillaries, which we're gonna look at in a second. This doesn't mean anything to you right now, and it shouldn't, okay? Once we are leaving from the peritubular capillaries, we come back out within the cortex, and blood is going to enter into the cortical radiate veins, okay? So we're going backwards now. So here is the cortical radiate veins. It's going into the arcuate vein here, okay? Then it drains back into the interlobular vein, and then there's no segmental veins, but we continue right into the renal vein and gets dumped into, and we've got it all listed here, into the inferior vena cava, because that's where the kidneys are going to be located on down. So it's got to be the inferior vena cava, right? So blood's going to come out this way. What I want you guys to see, and I know I've kind of muted a lot of you guys. I'm going to unmute some of you guys. Hold on a second. Let's see. Some of these are unasked to mute. Okay. So I muted you guys. If you want to talk in the chat, that's fine too. Did not mean to do that. But I want to show you now what the nephron actually looks like and what I'm talking about when I talk about the afferent, okay, or afferent arteriole to the glomerulus, to the efferent arteriole, to the peritubular capillaries, to the vasa recta capillaries. What do we actually see, these little nephrons that are found in the cortex? What does it look like? So we're going to actually show you, we're going to pull back up on this handout and show you an image. Okay, now here we are. So let me explain where we are. We are in the cortex, but here is the arcuate artery, creating the arch, arcuate vein, okay? Here is the cortical radiate artery, cortical radiate vein, okay? So that should make sense where we are. Down here is going to be the pyramid or medulla area. Up here is going to be the cortex of the kidney, okay? So as we are in the cortical radiate artery, what are we looking at when we're talking about the nephrons? Okay, so we entered from the cortical radiate artery, then we talked about a afferent artery, okay? But now we are supplying the nephrons themselves, okay? This right here is one nephron. This right here is another nephron, okay? These are two types of different nephrons, but what I'm gonna do, this is the basic nephron. We're gonna talk about this one first, and then we're gonna talk about the differences that are in this nephron, okay? So here's the cortical radiate artery. Here's the afferent arteriole that's coming into this little structure here. This is the glomerulus. The glomerulus is literally just a capillary network, okay? It's just a capillary network. So from the glomerulus, what happens if you've got pressure, right, into this blood pressure, into this capillary bed, we're going to have some of it's going to secrete out of that capillary network and via diffusion, pressure, filtration. Filtration is the big thing we talk about with kidneys. Kidneys create urine mainly from filtration. That's what's happening here. Now, there are special types of cells called podocytes. Podocytes are like these little finger-like projection cells. They grab into the glomerulus and they grab tight and they create little slits, okay? We're going to look at those in a second. They're not, it's a better image than this. Let's put it that way. So what happens with those podocytes and the pressure and the filtration and all that happening, you're going to have some of the blood, okay, some of the blood, meaning it's no longer blood, right? Because some of it's being pushed through very small molecules in water, basically, is what we're looking at, pushed through into this little tubule. Okay, this is called a nephron tubule, okay? What happens is when we push that fluid into the tubule, we call it filtrate, okay? Because filtration has happened, and this is the filtrate now, okay? So this tubule is wrapped around with this little, you can't even tell what it is. It's turned blue, right? It's almost like a purplish blue. It's an in-between mix from here, okay, the arterial, which is oxygenated blood, and it's pushed, right? This is where we push via the glomerulus, the filtrate, the fluid through the tubule, but then we have this weird kind of structure, purple structure. Well, this is called the peritubular capillaries, and it is created via the efferent arterial, okay, the efferent arterial, which is creating this little section right here, and it's now branched off to create tubular capillaries by this point. I'm going to explain what they're doing here in a second. Now, this tubule, this nephron tubule, we divide it up and we name it depending on where we are on the length of the tubule. Okay, let me tell you, for example, we started from here, I'm following the tubule. This is all considered here, I'll keep following it out this way, okay? This is all considered the proximal, meaning closer to the proximal convoluted, because it's all convoluted, tubule. So we call it that's what we go here, the proximal convoluted tubule, okay? As we go through, and I don't know if I could really follow this through, it's really hard to kind of, oh, I see it, here we go. As we go through this proximal convoluted tubule and we have these peritubular capillaries wrapping all around it, this proximal convoluted tubule will eventually go down and it will descend, okay? This is the descending, okay? All right. The descending, what we call either loop of Henle or nephron loop, okay? So this is the descending, because it's going down, nephron loop or loop of Henle, okay? So the descending branch of the nephron loop or loop of Henle. This is the actual loop found here. We call it loop of Henle, named after the doctor, or the nephron loop. And then as we come back up, this is called the ascending, okay? Nephron loop or loop of Henle, okay? This is going to be the ascending, it's going up, nephron loop or loop of Henle, okay? Now, after we've come back up from where ascending, we're now going to be in the distal, we are classified as I'm trying to follow it, the distal convoluted tubule. Wow, I actually followed this whole thing through. Now, it's going to be the distal convoluted tubule by this point, all right? And I don't know that we have it really labeled here. You just need to be able to look, be able to know you've got the proximal convoluted tubule, you've got your descending loop of Henle or nephron loop, you have the nephron loop or loop of Henle, then you have the ascending nephron loop or loop of Henle, and then of course we have the distal convoluted tubule, okay? So I think you guys already kind of getting the point that we've created a filtrate via filtration, this fluid that's passed through into this tubule, and this tubule is going the distance, basically. It is going quite a distance, let's just say that. And now we've got these peritubular capillaries. So why the heck would capillaries be following this convoluted tubule that holds potential urine? What's the whole purpose of that? Okay, so there is a purpose of that. And I'm gonna talk about it here, then I'm gonna re-mention it because I've got it, you know, where we can talk about it specifically too, once again. But what is happening? It's kind of like it's a balancing act, okay? The balancing act through this tubule is we are going to have this filtrate come in, these small particles and water comes in. Then as all this filtrate comes in, we are going to balance what gets secreted, absorbed, all of that to create the final product of urine, okay? But it's a balancing act through this passageway of the tubule. We decide, do I want to get rid of water? Do I want to get rid of amino acids? Do I want to get rid of glucose, for example? So our body's constantly going through that thought of homeostasis. What do I want to keep? What do I want to get rid of? What's considered waste? What's vital? Because remember that homeostasis means I don't want too much of something because it could throw me off. It could be dangerous. Or I don't want too low of something. It could throw me off and be dangerous. We don't want too much potassium, too much sodium. We don't want too low potassium, too low sodium, for example. So it's a balancing act. But let me talk about specifically what happens where in the tubule. Okay, so what happens? You create this filtrate. You're in the proximal convoluted tubule by this point. You've got all these peritubular capillaries wrapping around the tubule. What happens is there's a term we use, we call it reabsorption. Okay, so we've already created the filtrate. Reabsorption is when we take certain things away from the nephron tubule and we put it back into the blood via the little peritubular capillaries, okay? What kind of things are we going to grab from the nephron tubule? What kind of things are important to us? One, it could be water. It could be things like glucose. It could be things like fatty acids, small amino acids, okay? Things that are very small that are vital to us that we really need, okay? So those are gonna be like immediate things that you're gonna grab because we need them, okay? Now, let me say this before I continue on, because now we're in the proximal convoluted tubule, we're continuing on. Let me just mention that bigger proteins should not be passing through the glomerulus. The glomerulus should be in a good enough structure, not damaged. A good healthy kidney should keep bigger molecules out. Okay, we'll talk next week about the kidney failure and what all happens, right? So small things are going to go into and create the filtrate. The proximal convoluted tubule, we're going to take small amino acids. We're going to take things like that, not big proteins, actually folded proteins, right? We're going to take glucose. We're going to take water. Those kind of things are going to be reabsorbed back into the peritubular capillaries. As we go through this tubule, we come down, I'm not going to follow it all the way through, but here we go. We come down into that loop of Henle. The loop of Henle is wonderful for things like grabbing water, okay? All right, so it can concentrate urine to some degree. So we can hold on to the water, okay? And then the solutes are going to be heavier and they're going to continue through, okay, with some water through the tubule to the distal convoluted tubule. Now, things that happen in the distal convoluted tubule, what's going to happen? Because now we've reabsorbed all this stuff. We've gone all the way through from the proximal convoluted tubule to the loop of Henle. Now we're coming back up. We've absorbed all this stuff. Now here comes a more so of a balancing act. What we do in the distal convoluted tubule is we are going to secrete out things that we've overly absorbed. I know that sounds crazy, but we're going to secrete back out some things. It could be things like maybe we never got it into the tubule. Maybe we need to diffuse some things via the peritubular capillaries, things that we need to get across. We need to keep okay, into the tubule to be flushed out, to be urine. Things like drug metabolites, urea needs to be in there. If we take back up urea, we need to get rid of it. Uric acid, okay, creatinine, right? So all of these things need to get rid of. We need to get rid of that waste. If we have electrolytes where they're over, like you've got too much potassium, got too much sodium, for example, we need to get rid of some of it, right? So when you're in the distal convoluted tubule, we are going to secrete back out into the tube from the peritubular capillaries to go into the tube stuff that we don't want. Okay, so here's that balancing act. And once you've created that balancing act where we finally decided this is what's going to be left as urine, it's going to come from the distal convoluted tubule and it's going to come down here into something called a collecting duct. Okay, this is a collecting duct. Now keep in mind where we are in the actual kidney. All right, this was the cortex up here. This is that the arcuate artery, okay? This is that arcuate vein, for example. So the medulla is down here, okay? For this image, for what we're looking at, just knowing what I know, I would know that the pyramid, okay, a pyramid is found here. And if you notice, this collecting duct goes on down. Okay, so we're going to explain, I'm going to explain where that collecting duct, what it looks like from a large scale, okay? So that's a collecting duct. The collecting duct is going to take the formed urine by this point, what we've created as potential urine at this point, and it's going to go to the apex of the pyramid, the papilla, and it's going to dump now what's created as urine into the minor calyx, then to the major calyx, then to the renal pelvis, then to the ureter. But we're going to look at it, that other image we looked at, we're going to come back to it, okay, of the kidney cross section. What's happening over here? Let's talk about it. I need to move you guys. Hold on. Okay. This is called a cortical nephron in the cortex. This over here, this one that just looks so different, right? It jumps down. This is called a juxtamedullary nephron, okay? So what is happening in the juxtamedullary nephron is part of it, yes, is in the cortex, but a good portion, that loop of Henle, extends all the way down into the medulla, okay? Now, all the structures are the same except for one thing. You've got one, there is a longer loop of Henle, okay, or nephron loop, and the peritubular capillaries are no longer peritubular capillaries. We call them the vasa recta capillaries. Vasa, right, the vessels, meaning recta, meaning straight, okay? So they are straight vessels that go along the loop of Henle or nephron loop. The whole purpose of this type of nephron is they help concentrate urine, because the loop of Henle is going to pull water, okay? Mainly pull water, okay? And so you're going to leave with concentrated urine. So you've got nephrons that are in the cortex, the cortical nephrons, and then you have the juxtamedullary nephrons. All the structures are still the same. For example, here we go. We've got the afferent arteriole, the glomerulus. This is something I need to name for you guys. This, because you can see it on this one a little bit better. What's wrapped around the glomerulus is technically the actual shell. The start of that tubule is called the Bowman's capsule, okay, or glomerular capsule. You can use both of those names, Bowman's or glomerular capsule. Here is, I can't even see where it ends. Maybe this is it. This is the start of the proximal convoluted tubule. Here it continues. Yep, this is it. Now it's going descending loop of Henle. It's going to create the loop. It's going to go ascending loop of Henle or nephron loop. This is the distal convoluted tubule. Here it comes. Here's urine dumping into the collection duct. Okay. All right. And you guys can, I know it's hard sometimes for me to be able to see when you message little questions in there, but if you have anything you want to put in as a question, go ahead and do so before I get too far past this, because I know it's difficult. Let me go down to that other image. And this is just information, by the way. The renal corpuscle, that's the glomerular Bowman's capsule, excuse me. So talks about the filtrate. Cortical nephrons are 85% of the nephrons of the kidneys. You have the efferent arterial supplies, peritubular capillaries. We already talked about that. Juxtamedullary nephrons produce concentrated urine, okay? And they have the vasa recta, okay, instead of the peritubular capillaries. Now, come back to this image. Something I want to show you on this image that they denoted on this image, they made a good point to show it, is here is each one of the pyramids, okay? This is a pyramid, okay? This is a pyramid. This is a pyramid, right? Do you notice that there's striations inside of the pyramid? Hold on. The striations in the pyramid are supposed to represent the collecting ducts that come through the pyramid. So we are looking at the nephrons that were up here in the cortex of the kidney, right? Now, here, the striations are supposed to be the collecting ducts, right, of this nephrons as it comes down. Now, here's the papilla, right, or the apex of that pyramid. When the collecting ducts come down, they dump urine here, okay? This is the minor calyx, major calyx, and urine's going to flow through down into the renal pelvis, down the ureter, okay? Off to the bladder it goes. So if you come back to this image, you'll be able to see when you follow the pathway, you're able to see, here we go. Now we're considered the nephron, it's boxed off, the afferent arterial glomerulus, the efferent arterial peritubular capillaries or vasa recta capillaries, and then now cortical radiate veins, right, returning blood, arcuate veins and back, okay? That's the whole basis of how I wanted you to understand it. See on a big scale, see what the nephrons actually look like in the cortex. How is blood actually creating urine, and how does urine get out of the kidney, right? Okay, so let's move. I know I keep moving the wrong bar. Let's move up. I think that's the direction I wanted to go. Can't really remember. That's really sad. Let's see. Let me move this down. I gotta move my bar over. Okay. Now, we've looked at the nephrons. I don't want to miss anything. I want to go in order the way I want to go to order. Here we go. This is what I want you to see. Sorry, my screen is not huge and everybody can see all this, but this is a really big image of the glomerulus if you haven't figured that out yet. All right. What I want to make points of showing you off of this image. One, okay, obviously here's the glomerulus, here's the afferent, you'll have to label these afferent arterial, here's the efferent arterial leaving, right? This is coming, this is going, right? Now, here is the Bowman's capsule, okay, glomerular capsule. Here's the start of the proximal convoluted tubule. You've got filtrate that's going to be inside of it, right? Inside the lumen, inside of the tube. Now, you can see the red blood cells. Those are representations of the actual capillary network, right? But then you see these little cells here. These are the podocytes, and the podocytes have these little finger-like projections. And what it's doing is it's grabbing onto these little capillaries and creating little spaces, right? So it can grab and open up to allow for better filtration to happen. That's the whole idea. So you've got the pressure that comes in, the afferent arterial into the glomerulus. Now we're creating filtration to happen, the pressure, just like we've talked about filtration before. You stick your finger on the water hose, for example, and you have it on, you can only hold it for so long, and some of it's going to seep through. Well, that's the same idea as filtration here, okay? So you're going to have some of that, part of that blood filter out here, and here we go at the balancing act for homeostasis, decide what's going to go, what's going to stay, for example. So what's going to stay and be excreted, what's considered waste, and what do we need to go push back into the circulatory system, right? You can see the little cells that are wrapped around the little blue ones too. We're going to talk about that also. So something that you need to label and understand, which we'll talk about in more detail in a second. You've got a little blurb about it below. But these are called, two different terms, granular cells or juxtamedullary, juxtamedullary complex, for example, cells or granular cells. Left, far left. What is the structure on the far left? Let me see. Oh, this. I haven't gotten there yet. I'm going to get there, though. It's a good question. So these are, I will mention it in a second. This is the afferent arterial. These are granular cells. what it's doing is it is literally picking up on the amount of pressure that's coming in to these afferent arterioles, okay? Now, it works with these blue cells. These blue cells are attached to this structure. This structure is actually, believe it or not, is the distal convoluted tubule, okay? It's come back up and it's come next to the glomerulus. And what it's doing is it's picking up on the amount of salt, sodium chloride, that's found or that's been put into the tubule, okay, what's going to be eventually urine. So it's picked up on salt, and these two, the granular cells, and these are called the macula densa cells found on the distal convoluted tubule, are going to balance how much pressure, how much pressure for these cells, these granular cells, should go into the glomerulus, can't talk all these words sometimes, into the glomerulus. So it decides how much pressure. The pressure will help one of two things. Let me explain this. So these are the granular cells, which work with these cells. But the granular cells are going to decide how much pressure should come in. But when we decide how much pressure should come in, it allows for the control of how much pressure gets pushed in and created into the filtrate. So how much is going to be lost, right, into that tubule, and how fast it goes through that tubule. That's another thing. The faster it goes, the less we can do the balancing act and more gets pushed out. For example, more gets lost than maybe we want, or more, for example, doesn't even have time to secrete back in to even create part of that urine, okay? So maybe we wanted to reabsorb sodium. Maybe we wanted to get rid of some potassium, for example, I don't know, whatever it may be. But if we go too fast, then we can't really do the balancing act, for example. It's not going to be a proper homeostasis, right? Because it's too fast to go through to keep balancing. So these granular cells are going to help with that. However, they do work with these cells. These are called the macula densa. This is the distal convoluted tubule that is where it's found, and it wraps up next to it. The reason they are made into around the glomerulus and next to and part of the distal convoluted tubule is then by the time we're in the distal portion of the tubule, we need to evaluate how much sodium chloride is there, because when we evaluate how much sodium chloride is there, sodium chloride is also functional with blood pressure. If we lose too much sodium, then water goes with it, for example. So we talked about that with osmosis, for example. So water's going to follow the higher solute concentration. Remember, we talked about that. But when we do this and we balance and we pick up how much sodium is lost, and we correlate these cells, or I say correlate these cells, work with these cells both together, we'll notice that you guys have probably heard of the hormone renin, but we'll notice that these two types of cells like to work together because they will tell the body whether we need to secrete out renin, okay? And renin is going to help with what we absorb, blood pressure, all of that. Something that you guys didn't really, you talked about vasopressin. That was one of the things that you did talk about with the conditions. Renin helps and works with vasopressin, for example. So it's a balancing act to help with blood pressure, so you're not losing too much water, because if you lose too much water, then you're affecting your blood pressure, okay? And for example, these granular cells is also helping with the balancing act, the homeostasis, not just blood pressure, but some level of homeostasis too, okay, in general. Come down here, it talks about these granular cells, juxtaglomerular cells. They are mechanoreceptors, meaning physical motion will cause them to decide what to do. So the granular cells are going to help with blood pressure. Remember, I talked about how much pressure comes through. They work with renin. Renin helps with the blood pressure. If it's in function to, if you have a drop in blood pressure like this talks about, okay? So we want to keep it balanced, that homeostasis, not just with electrolytes, certain things we want to absorb back into the bloodstream, but also we want to work with, because it's a filtering system with water also, and electrolytes because of or not just the homeostasis, but the osmosis theory with everything, right? We want to balance with blood pressure too, okay? Macula densa, there are chemoreceptors, okay? Sense the sodium chloride concentration in the filtrate. So for example, high sodium chloride in the filtrate causes vasoconstriction, hinders the blood to the glomerulus. You don't want to lose too much, right? We want that sodium chloride to be reabsorbed. Same thing, works with the renin for blood pressure, okay? I know I just spent a lot of time on that, but anyway, it's neat, it's cool. So I think it's valuable time at least. All right, with that said, let me come down here because I think I've gotten all the little images. Did I get all of them? I did. There's one more image I want to show you guys that I have on the handout. Oh, not on the handout, but on that's not on the handout, for example. But let me make sure I've gone through everything real quick, because I know I skipped through just to go in the order that I like, because it makes sense to me. Oh, here we go. I was like, there's something left, I know. Okay, you know how we talked about the whole peritubular capillaries, the creation of the filtrate via the filtration in this capillary network called the glomerulus. So basically you're putting pressure on that capillary network, you're having some of that blood fluid pass through into the tubule, right? We talked about that to create the potential of urine, right? What's going to be theoretically urine eventually. So urine formation results from 3 processes, right? We talked about this. Filtration is one, when we put pressure against that glomerulus, and then just like your finger over a hose, some of it's going to seep through. So that's one that creates the filtrate, okay, the beginning. And then we have reabsorption. So come down here, let me show you. So then reabsorption is going to happen this way. That's number 2. That's reabsorption, so you can see it. So reabsorption is going to absorb, we already talked about this, water, things like water, small amino acids, glucose, for example, maybe fatty acids, things we want to grab back up, right? Sometimes we over reabsorb things. And then we have, secretion, okay? So filtration is the role of the glomerulus, where the portion of the blood is going to pass through the glomerular bed, and glomerular capsule creates the filtrate. Filtrate enters the proximal convoluted tubule. The tubule reabsorbs, okay, where the filtrate component is going to move from the tubule to the blood peritubular capillaries, right? And with a little note here, some is active, some is passive, just like we talked about when we talked about, you know, movement of body fluids. We talked about those examples. And then we have the secretion at the end. Okay, secretion is a reverse of reabsorption. So it's where the substance from the blood or peritubular capillaries are going to go back into the nephron tubule to become filtrate, right? To eventually come urine. So here is going to be the filtration process. The filtrate's going to happen. Here we're going to have some things reabsorb in the proximal convoluted tubule back to the peritubular capillary. Then we're going to have in the distal, okay, convoluted tubule, you're going to have secretion come back in. Okay, so we're going to secrete things back into the nephron tubule. So things like, think about this, maybe we need to get rid of certain electrolytes that are too high, too low, for example, or whatever, too high. Maybe we need to make sure that the urea, we get rid of all the urea, the uric acid, creatinine, things that need to be waste, right? So we dump it back if we've over reabsorbed, for example, okay? So maybe we have too much of something. Then finally, after we've done the secretion portion, we finally create urine. It goes via the collecting ducts down the pyramid to the papillar apex of the pyramid, to the minor calyx, to the major calyx, to the renal pelvis, then to the ureter, then to the bladder, right? So this is a good little image just to keep you on track with that. I like that little image, it's good. The other thing I want to show you from a big scale, I'm about to hop off this and reshare my screen. I apologize, I know it's going to take a second. Let me do this. Hold on, just bear with me. I know it's off. Let's see. This one. Okay, you guys can see this, right? I hope so. Okay, the other thing that I want you guys to see is where do the kidneys actually sit? So this is something, and I know you guys will hit this in lecture too, but this is something I think you guys may not realize, so I just want to go over this. So we're looking at a cross-section of the abdominal cavity, for example. So here is the peritoneum here, and then behind it, so you guys remember the peritoneum is going to hold all the, you know, all of those organs in the front and that serous membrane, right? You got all your guts, you've got your liver, you've got all these different, you know, organs that are up here that is going to be found in the front, right? But the difference with the kidneys is the kidneys are called retroperitoneal because they are behind the peritoneum. Okay, they sit, and this is what it's trying to show you. They sit behind the peritoneum. They have a lot of fat that sit around them, okay, and they attach to the back wall. If you look here on this image, this person is turned facing me or facing you if you're looking at the screen. For example, this is the right, this is the left. One of the reasons I know that, and the only reason I really know that off this image is the right is always a little lower than the left. The liver's also over there. It takes up some space, so it pushes on that region. So the right usually sits a little lower than the left. So just keep that in mind. I just wanted you to be able to see this because I know that's something that, you know, some students are like, what do you mean retroperitoneal? What does that mean? It's behind the peritoneum, okay? Or the peritoneal cavity. Okay, now I'm going to go back to, I have to reshare you, so I'm mindful of it. Hold on. Let's see. We're back. Okay, I'm going to go back to this screen, and then now what I want us to do is I want to come back to exactly what I want you to do, because now we've talked about the objectives to some degree, right? We haven't hit all the objectives. We've hit the anatomy of the kidneys, where they sit, how does urine go through, what are the basic physiology of that homeostasis of balancing to decide what's going to be in the blood, what's going to be in the urine kind of thing, right? And we talked about the structures. So we've talked about some of this off of the objectives. However, we've not gotten to the lower portion of the objectives, which we're going to talk about next week. So I talked about meaning the pathophysiology, what happens when the kidneys don't work, for example. Okay, what happens when you test someone's urine and you see levels that are not normal? Okay, what does that mean? We're going to talk about that next week. You'll have the urinalysis case studies to do after we talk about that next week. It's not crazy, but it's not anything like, it's not a ton, let's put it that way. But once you get this down, it will help you next week. Here is the dissection, or the cadaver tool dissection, anatomy physiology revealed. Like I said, we already talked about this. Make sure you go through all these images, especially after I spent time talking about it. Please make sure you go through all of these images, because now you have something to compare after we talked about the handout. What these actually look like. You know, some of these you guys know are actual images, some of them are not, some of them are animated, but most of them are actual images, which sometimes throw people off. But even everything down to like the renal corpuscle, glomerulus, everything, the Bowman's capsule we talked about, peritubular capillaries, vasorectal capillaries, all of this is things that you need to look up. So with that said, you've got every single thing that I talked about is going to be in that anatomy physiology revealed dissection tool. Come down here, then you can look at the kidney, and then you can actually see like the urinary tract and break them up, female, male. You should be able to look at those. And then of course, here's the practice on the nephron, just for you guys to color, label, all that. Like I said, you don't have to do that. I hate to say it, but it's not required. And then we've already talked about the dissection, okay? Dissection video on what to do. Dissection video is going, like I told you, it's going to be every little, once you get past setting everything up, go down here, and every little thing, like small piece of, you know, like label 1 through 7, name and date next to it. Each photo needs a card next to the kidney giving a name and date. Those are going to count for points, for example. Each photo needs to be clear. Take one photo for each identification item, for example. You know, all of these little things that we've put on here, they actually directionally count for points. I know that sounds crazy, but they do, okay? Even it's not as basic as just label these things, you need to do every little thing accurate. So all photos need to be in these type of format. Okay, put it in Dropbox. Don't just send it to us, right? And then I'll go through and grade them. That is done the 25th, like I said. Don't touch this yet. We'll get to it next week. And then eventually you'll get to the post-lab urinary quiz. So if you get confused, go back up to due dates, look when everything's due. If you look at week 9 and you don't see the urinalysis stuff, it's because it's not due yet, because we haven't gotten there yet, okay? All right. Any questions before I leave you guys? Before I go, you're free, go look at these items. You've got time to do your dissection. Decide a good time to do that. It's probably not tonight. I don't know. That's up to you. But go through what I would do tonight. Spend your time, go in through and look. Now that we've spent time looking at the handout, talking about the images, how everything works, I would look at all the different structures you need to know. Now that you understand what these structures and generally do, and the next week we'll get to the pathophysiology, the urinalysis, what things actually occur when things don't work right? So, and then what ranges and things like that with testing. So do you guys have any questions before I leave you tonight? And I think I've got some of you on mute. Hold on. So you guys can type it in. I can hit you guys as unmute. Any questions before we go? Okay. I assume nothing. If you guys know how to do little emoji icons, send me a thumbs up if you're cool with everything, you understand what you need to do for the rest of the evening. Awesome. Thank you, Zach. I know some of you are looking for the little emoji con things, but if you're done, I'm gonna, if you guys have any questions, you guys feel good about everything, then what I'm gonna do is you guys can email me, text me any questions. If we need to set up a separate Zoom time, we can do that. If you have any questions in particular that you want to ask one-on-one, I've always been open to that too. So otherwise, if you guys are good, I'm gonna let you go, jump into those activities. If you have questions about the dissection, like I said, text me, email me, whatever you may need, and I will help you through it. All right. Later, guys. Bye.